So we're going to be covering metabolism, and 2.8, 2.9 are going to be a part of this because 2.8 and 2.9 are basically the S cell precursors to respiration and photosynthesis. So we're going to start with metabolism. So metabolic pathways. So the fundamental thing to know about a metabolic pathway is they depend on exergonic and endergonic reactions. Exergonic can also be exothermic. Endergonic, it can be endothermic. That's usually how it is. So the metabolism is a sum of all of the anabolic and catabolic reactions in an organism. Most of the chemical changes happen intracellularly within metabolic pathways, and these are facilitated by a bunch of different cascade reactions, different pathway reactions with different enzymes at specific times, controlled by specific things and environmental factors. So enzymes are going to be the key thing to these pathways, because what they do is they lower the activation energy needed for a reaction to occur. So they make it easier for a reaction to occur, or they can speed up the reaction. In the sense that a reaction can be spontaneous in that it will naturally occur, but it might take a really long time for it to naturally occur, so it will speed it up. So part of understanding enzymatic function is in understanding how inhibition works. There's going to be two main types of inhibition. It's going to be competitive and non-competitive inhibition. So competitive inhibition, like you see here, is the main substrate that wants to bind to this enzyme, and this is going to be the active site. It would have binded at the active site, but a inhibitor bound to the active site. So they're competing for the same active site. So this is an example of a competitive inhibitor is Relenza, which is I believe an antiviral, while non-competitive inhibitors, they will not try to bind at the active site, but they're going to bind at this place called the allosteric site, which is another site on the enzyme. And the binding to the allosteric site is going to change the configuration of the molecule and change its shape so that this substrate cannot bind to the active site because it doesn't fit anymore. So a really important example of a non-competitive inhibitor is cyanide. Cyanide is really deadly because it blocks oxygen being the last electron acceptor in the electron transport chain, so it can kill organisms through that. Feedback inhibition is a type of allosteric regulation. So it's negative feedback can control the activation and continuation of metabolic pathways. So this happens because, so you can see here, we're going through this pathway. When you have enough of the product will bind to the first step. We'll go back to the first step and bind to the enzyme in this first step and then block that from making more product. So it's a nice way of self-regulation. And then the substrate won't go through all of these pathways. And if you see, there's a lot of enzymes in this cascade here. So it's going to be using a lot of energy. So this makes sense. If we have enough products, the product will block more energy from being used. An example that I believe you have to know is, or a useful example would be isoleucine. It's used for the biosynthesis of threonine. That's catalyzed by threonine deaminase. Isoleucine can bind to the allosteric site on the enzyme. Now we're going to transition to talking about kinetics. So there are going to be three main factors that affect enzymatic function and activity. It's going to be pH. Here is just, you want optimal pH. It's really, you don't want too much in the either direction, just want in the middle, because if you have if it's too acidic or too basic, it's going to interfere with the active site and cause denaturation, which can affect the secondary and tertiary structure of an enzyme. Temperature here, you don't really want either in either direction, but you can see here that the function is a little better when it's a little more heat. So a little more heat is actually really good for an enzyme. This is just because adding heat to a system increases the amount of collisions. So more collisions happening means it's more likely for an enzyme to collide with its for particles to collide with the right configuration for a reaction to happen, and you have more heat, so there's more energy being added to the system. So you have more of that needed energy for a reaction to happen. And substrate concentration here is just looks pretty linear up to this point. The more you add, there's a direct correlation with how much the rate of reaction speeds up, and then it sort of tapers off here, and this has to do with the availability of the enzyme. Past here, there's going to be more substrate than the enzyme can like deal with at a time, so adding more at some point just doesn't do anything. You just have to wait for the enzyme to finish what it's doing and then move on. So you have to note what the rate of reaction is. It's just s to the -1, which is like 1 time divided by time taken. And you have to also be able to like distinguish the types of inhibition on a graph. I've seen past paper questions that are like similar to this, where you have to infer what type of inhibition or like what type of reaction it is based on the curve they give you on a graph. So I would know that. So then we're going to move on to cellular respiration. So cellular respiration is dependent on the fact that redox reactions happen simultaneously, so it's oxidation and reduction. When I was in IB, I learned oxidation and reduction as oil rig. So like oxidation is loss, reduction is gain. So oxidation, you lose hydrogens, you lose electrons and energy, but then you gain oxygen. And reduction is gain because you gain electrons, gain hydrogen, and energy, but you lose oxygen. So the important molecules you really have to know for this reaction, for the redox reactions in cellular respiration, is going to be NAD, NADPH, and FADH. These easily give and accept electrons and hydrogen plus. They're going to be your electron and hydrogen carriers. I also want to point out that it can be confusing if like something is being oxidized, but it's also referred to as the reducing agent. So remember that like if something's being oxidized, it's reducing something else, and then if something is being reduced, it's oxidizing something else, so it's the oxidizing agent even though it's being reduced. So there's going to be two main types of respiration. There's going to be anaerobic and aerobic. Anaerobic means you don't need any oxygen. It has a smaller ATP yield. It happens in the cytosol of a cell, so you have a 4 net yield. Aerobic, you require oxygen. Anaerobic would be just like glycolysis, and then anaerobic would be like everything from the link reaction to Krebs cycle to electron transport chain. I've seen varying numbers on the yield, but it's the one I've seen used a lot is 32 to 34, but I've seen as low as like 28 to 34 or 28 to 32. So one application of this is the IB wants you to know how respirometers are used to measure cellular respiration based on the amount of CO2 or O2 in the atmosphere of this like little apparatus. So it's affected by different hydration, light, age, and other factors. So you need to be able to analyze the results of a respirometer experiment in germinating seeds or invertebrates. So if they give you the data, you'll have to be able to analyze that. I haven't really seen anything like that on past papers, but it's in the syllabus, so you are required to know it. From glycolysis, which was that anaerobic process, you either can go through fermentation or aerobic respiration, depending on how much oxygen you have in the air, or if you're an organism that even requires or wants to use oxygen.
কতক্ষণ লাগবে কয়েক পরে কালকে
Fermentation produces alcohols and carbon dioxides in yeast and plants, while fermentation in animals uses makes lactate. So fermentation is going to be like those things responsible for making all the beers, all of the sugar molecules that we like to eat. Fermentation is a really important process. Lactate production in animals is pretty toxic for us, at least, mainly because of lactic acid, I believe. But this is like a pretty helpful diagram to see where steps of the pathways are and where it happens. You also need to know where these parts of the pathways happen. So glycolysis is going to be the really main thing that happens in the cytosol of a cell. And then mitochondrion is where most of everything else happens. Pyruvate oxidation, citric acid cycle, oxidative phosphorylation, and etc. So glycolysis is just the breaking of sugar. Glyco means sugar, lysis means breaking. So aerobic respiration occurs if you have presence of oxygen and you make water and carbon dioxide and a lot of ATP. So here you have this like 6-carbon molecule glucose, and you need an input of 2 ATP. That input of 2 ATP and the breaking of that phosphate bond in ATP to ADP will catalyze the reaction for it to become fructose disphosphate, and then that is a really unstable molecule, so it will split in half and create G3P. The IB can also refer to it as like triose phosphate because it's a sugar and has a phosphate, so you do have to know that it can be referred to as triose phosphate. But I think it's a really stupid name, and I really hate that. I've never heard it's called triose phosphate like anywhere else. Like maybe it's in an odd paper or two, but like calling it triose phosphate is a really dumb name. So don't expect to hear it called triose phosphate anywhere else but in IB biology. Like as a biologist or like as a senior majoring in like double biology majors, never heard of triose phosphate. It's always been G3P. So when you have these two G3P molecules, you need more input. So you need this NAD that we talked about earlier, and then you need ADP, and that will this pathway here will create 2 pyruvates. So remember, the input of these 2 ATPs here is the energy investment phase because you're investing 2 high energy molecules for that, mainly just for that breaking. Guys, like please freaking mute. It's very annoying to be constantly interrupted.
Sorry. Pô, sai daí, mano. É muito mais impactante a gente parar a fantasia de Minions e.
Where was I saying? So here's the energy investment phase. So you minus 2 ADP, and here is creation of 2 ATP at the end of this for each. So it's 4 plus 4 here, minus 2 here. So you have a 4 yield.
Sim, a gente fica parado. Essa porra ligação.
I'm server muting you because it's a little bit disruptive. I'm really sorry. If you have questions, like just ask them in the chat, but like it's really disruptive. You have your investment phase here, so you have you're putting in 2 ATP, so minus 2 ATP here, and you're getting 4 ATP total here. So 4 is the yield for the ATP, but it's actually net 2 gain because net factors in the amount of ATP used initially. And this pyruvate is going to be used later for another pathway. So remember, glycolysis produces glycolysis 2 ATP to make 2 G3P, and then those G3Ps become pyruvate, and at the end you have 2 net. So you need to know energy payoff, energy investment phase. You need to know what G3P is. You need to know the pyruvates. You don't need to know, I don't think you really need to know fructose disphosphate. But yeah, you need to know, also remember that G3P can be named as triphosphate. So after you finish glycolysis, you have this pyruvate, and you're going to go through this part for the citric acid cycle. Usually I see the citric acid cycle as like being a part of like coupled with pyruvate oxidation, but it's whatever. It happens all in the mitochondria, so it's going to be transported from the cytosol to the mitochondrium. So the link matrix, the link reaction is going to be still in the matrix. So these pyruvates here are going to be decarboxylated, and then that carbon gets released as carbon dioxide. So this is where part of the carbon dioxide comes from as a waste product in respiration. So during this is going to be your pyruvate oxidation with acetyl-CoA. You do need to know acetyl-CoA. This provides the energy to reduce 2NAD plus to 2NADH. This NADH, remember, is going to be a high energy molecule because you're having energy and electrons and hydrogen stored here. So NADH is going to be a product we want to have. And then the 2-carbon molecule acetyl gets attached to CoA to make acetyl-CoA. Acetyl-CoA is going to be an important part in the citric acid cycle. So the Krebs cycle can also be called the citric acid cycle. You really don't need to know create the specific steps. Like this is way beyond what the IB would ever ask of you. You just need to know pyruvate, acetyl-CoA, and then like multiple inputs of NADH, NAD plus to get out high NADH molecules and carbon dioxide as a waste product. And you also get some ATP production. And then just that this cycle is regenerative. You do not need to know like all of these crazy names like also like succinyl-CoA or like succinate. Yeah, you just need to know like broad details about it. So here we go to the ETC, which is going to be in the cristae. So you need to know where each process is specifically in the mitochondrion. This is going to be in the cristae, which are like the folds in the mitochondrion. You don't need to know the complex names. My teacher, for whatever reason, made me know the complex names, but I am pretty sure you do not need to know the complex names. You just need to know that this NADH and like some of the energy here, you don't need to know succinate like in detail. You just need to know input from the citric acid cycle, such as NADH, is going to be used for the ETC. So NADH is going to be oxidized to NAD plus, and there's going to be like multiple redox reactions. At the end, you're going to have oxygen be the final electron acceptor, and that produces water. So this step here is actually why cyanide is so deadly. Cyanide blocks this step from happening, and that produces a lot of toxic. That's why it's so toxic. So what you really need to know is you have O2, you have FAD, and you have NADH, and that all becomes water. I would usually put a range like 28 to 34, somewhere within that range, because reactions aren't perfect. And then FADH2, which all goes back to regenerate some of the products in the Krebs cycle. I think with the Krebs cycle, you just need to know how much carbon each has. You don't need to know like names like alpha ketoglutarate. So we were talking about like different parts of the mitochondria, like the matrix and the cristae. So here's the cristae exactly. It's these portions right here. The matrix is different. It's within these little like folds, but the cristae is the fold itself on the outside. You really need to know. Really, the matrix and the cristae are going to be your most important parts. So the inner membrane space, you also need to know the inner membrane space. So the inner membrane space right here is pretty small. So you're trying to like make all of these folds, and it's really folded like this in the structure to maximize the surface area. And so that when the surface area is maximized, it can accumulate protons and make a gradient. If you know anything about chemiosynthesis that the IB wants you to know, you always want like a proton gradient so you can pump protons out, and then the protons will come rushing in, and that will help produce energy via ATP synthase. So within the matrix, you have the pH needed and the enzymes for the Krebs cycle, and then the inner membrane hosts the ETC and ATP synthase. So we're going to move on to photosynthesis. So a defining part of photosynthesis is how high energy wavelengths are. So plants are going to be harvesting light, visible light, along Roy G. Biv. Roy G. Biv is just the different colors, red, orange, yellow, blah, 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 blah. So chlorophyll is best at absorbing this like red and this blue light here, and it reflects green the most. That's why plants are green, because green is this like wavelength that they don't really care about, so they reflect it. There is a strong correlation between the absorption and the action spectra. The strong correlation occurs because if you look here, they have the same regions where like two main peaks happen, right here, and one where the main trough occurs. So like at this what that means is like at this wavelength, this like green yellow kind of area, this is where photosynthesis is the least productive. But here in this violet blue or like this red region, this is where photosynthesis is the most efficient. You will have to know like I don't exactly know if you need to draw it, but you do need to know that where exactly the peaks and the troughs roughly are. So like in this like 400 to 500 region here, and in this like 750, 700, 650-ish, 650-ish region right here for both of the spectra. It would be helpful to like draw it, but it might not be something that's necessary. But know the overall trends and how they correlate to each other. Okay, for limiting factors, these are going to be the things that really affect the effectiveness of photosynthesis. So temperature, light intensity, and carbon dioxide concentration. So temperature, this is mainly because enzymes have optimal ranges where they work the best. If it's too hot, it might start denaturing the enzyme. If it's too low, crystallization occurs, or there's just not enough energy for reaction to happen. Also, temperature has to do with the fact that a plant wants to keep it at a good temperature, so there's not too much water loss. Water loss is going to be a huge thing for plants. I think there's like other things you have to know from like other units about what I'm talking specifically about water loss or like adaptations in plants physiology. You do have to know about the waxy cuticle, but that's besides the point here. Temperature is really big for them. light intensity, the more light there is, the more photons that they can use for energy. So like those photons will be photo excite molecules in the cell, and they can use those molecules to provide energy for them. And it's up to until a certain point, because the light harvesting complexes are going to be all busy. When it's kind of like the similar thing like Vmax, when you have like when you add like substrate up to a certain point, and then the more substrate you add up until a certain point, it doesn't really affect it anymore. It's kind of like this concept. The more there's going to be general correlation with light up until a point where increasing the light intensity doesn't do anything, because the complexes are going to be all busy. Then this is also going to be the same principle with CO2. CO2, you're going to need this for the Calvin cycle, which is the light independent cycle that occurs mainly it really occurs during the night for a lot of plants. But yeah, this is for carbon fixation, where they turn carbon dioxide into sugar molecules. This is like one of the parts in the syllabus where I feel like I just kind of threw it in, but I do have to cover it. This is like a standard. So oxygenation of the early Earth. So early Earth oceans had a lot of liquid iron, and that iron would combine with dissolved oxygen to form iron oxide. And when the supply of iron in the ocean was nearly completely consumed, atmospheric oxygen began to accumulate. And before that, the early Earth was anoxic, which means it had really low amounts of oxygen in the air. And then when you started getting a lot of atmospheric oxygen, that was when one of the biggest mass extinction events in Earth's history occurred, because most of the life at that time was adapted to low oxygen environments. When you started getting a lot of oxygen, that killed off those life forms, mainly because oxygen has like a tendency to form these like really reactive oxygen radicals, like superoxide or hydrogen peroxide. That's why I use hydrogen peroxide to like sterilize an area, because the hydrogen peroxide will kill a lot of the anaerobic bacteria that could cause you harm. That's also why a lot of bacteria have catalase. Those bacteria that can live in oxygen have catalase to break down those hydrogen peroxide. But yeah, the reactions between that dissolved iron and the oxygen created like these really cool banded formations in different rock formations. And if you look through like different layers, you could see that happening. So for photophosphorylation, non-cyclic photophosphorylation, this is going to be happening in the stroma. This is going to be happening in the thylakoid. I'm sorry. This is going to be happening in your chloroplast. So I don't really think you have to know the in-depth details of this. But I had to learn it as an IB student, but I think my teachers made me learn unnecessary details. But you do have to know there is a cytochrome. There is like pumping of H+, so pumping H+ out of here. And since that concentration gradient of H+ is so high on the outside, what it wants to do is naturally diffuse in. And when it naturally diffuses in, it provides energy for ATP synthase. And ATP synthase is like this. ATP synthase will take ADP and inorganic phosphate, and then that energy provided by those H pluses flowing in after it's pumped out will provide the energy to make this into ATP. You also have to know about NAD plus and H plus here. But yeah, there's not a lot of detail that I think you have to know associated with this. You do have to know that light photo excites the pigment in the photosystem complexes. But yeah, that's about it. Don't try to bog yourself down with the details of like the specific names and stuff. Really have to know ATP synthase, have to know the H plus pumping and H plus flowing in, and how that works with ATP synthase, and you have to know this is happening in the thylakoid, and yeah. So with cyclic photophosphorylation, after you have photoexcitation, you have like a mobile. Oh my god. After photoexcitation, you have a mobile electron carrier that will not move the electron to any DP reductase, but you'll have it move it to the cytochrome complex to pump more protons in. You have cyclic photophosphorylation because the yields for NADPH and ATP are going to be different. You have this is because in non-cyclic photophosphorylation you make more ATP than you make ATP. So at some point the plant has way more NADPH than it knows what to do with and less ATP than it really wants. So you're going to go through cyclic photophosphorylation. You're going to skip this step here and you're just going to go from here to here, this kind of step here. The whole goal of cyclic photophosphorylation is just to make ATP. You don't care about making NADPH. However, also one thing to note is the electron did originally come from photosystem 2 and non-cyclic photosynthesis. So that's just how they connect in terms of cyclic and non-cyclic photosynthesis. Now you are moving on to the light independent reaction. So this was in the thylakoid, and it's going to be obviously the reaction that needs light. This is going to be a different part of this doesn't happen, I believe, in the stroma, and this does not need light. So you do need to know the details of some of this. You need to know Rubisco, and you need to know the short-lived 6-carbon intermediate. So Rubisco fixates 6 carbons onto 6 RUBP. You do need to know RUBP. And then it creates this short-lived intermediate that lyses into 12,3-PGA, or 12 3-carbon molecules, and then that's reduced into another 3-carbon molecule with 12 ATP. Also have to know this step because you're using ATP here, so there's 12 input here, and then you also have more triose phosphate here, but I don't know, I really hate the fact that they call it triose phosphate. I'm going to keep complaining about it. This molecule here becomes 12G3P by oxidizing 12 NADPH to 12 NADP+. And then 3 of those G3Ps will leave the Calvin cycle. 10 of those will go on to regenerate the 6 RUBP with some ATP use. But 3 of those are gonna two of the 3-carbon molecules are going to leave and make 1 sugar molecule. The math is kind of like a little bit weird because you might have seen it for 1 Calvin cycle, but I just did the math for 2 Calvin cycles because the whole goal is to make glucose. I just did it for 1 glucose molecule being produced. But yeah, you do have to know Rubisco, short-lived intermediate, 3-carbon molecule that's made into triose phosphate. You have to know the different amounts of ATP and NADP used. And then you have to know about the regeneration. So know the stages here, know about the regeneration. You don't have to know like the specific names of things like 1,3-bisphosphoglycerate, at least not when I learned this at the IB level. There might also be questions on the exams about comparing different metabolic processes. So between photosynthesis and respiration, photosynthesis, you need harvesting of energy, light energy, to photoexcite electrons across the thylakoid membrane. And then here you just get the electrons that are being moved from carrier to carrier that will reduce. In both of them, you're going to have protons being pumped across the membrane, but it's just different membranes. So the protons are going to be pumped into the lumen, and it diffuses into the stroma. And then the protons are going to be pumped into the intermembrane space in the mitochondria, and that diffuses into the matrix. These are also happening in different organelles. So here, this is going to be your mitochondria, and here, this is going to be your in your cytoplasm. Not cytoplasm. Oh my gosh. Photosynthesis. It's going to happen in. Okay, I just mentioned that. Thylakoid membrane, cristae, and this is photophosphorylation. Photo for light. This is oxidative. You require oxygen. Both of them require the pumping of protons, so you're going to need ATP synthase and electrochemical gradients for that. I think that's really the end of it. I will upload this to the revision website, and then I'll be posting practice problems questions from the past papers. So that should be like maybe tomorrow. It depends on when the recording is finished, but yeah.